Hello, my dear friends. Welcome back to Biology Explained. With various types of treatment going on for the cure of Covid-19, scientists have developed monoclonal antibody against Covid-19 for passive immunization of the patients with Covid-19. An article in Nature Communication was published by a group from Netherlands, and shortly after the publication, Israel announced that scientists at Israel Institute of Biological Research have successfully developed monoclonal antibody that can neutralize SARS-CoV-2. We will go through about what is there in the article, and we will also see what Israel Institute of Biological Research is telling about the monoclonal antibody. But before we go into the research paper, we should know what is meant by antibody and what is meant by monoclonal antibody. Antibody, also known as immunoglobulin, are Y-shaped protein, and they are produced by body's immune system to detect foreign harmful substance, that is antigen, like bacteria and virus. Antibody is produced by the B cells. B cells differentiate to plasma cells and ultimately produce antibody. The produced antibody binds to specific antigen to neutralize the antigen. As we have talked earlier, a typical antibody looks like a Y-shaped structure with 4 polypeptide chains. They have 2 identical heavy chains and 2 identical light chains, and they are connected by disulfide bond. There is a constant region and a variable region as shown in the figure. The variable region can have various structure and thus can bind to different types of antigen which invade our body. The constant region, on the other hand, has constant structure which interacts with the host cell. So, you can understand that these antibodies with specific variable region will bind to specific antigen and neutralize it, and the constant region is necessary to recognize its host cell. That is, a human cell will recognize a human antibody as its own part. However, a human cell will recognize a mouse antibody as a foreign part. Now that you know the basic structure of antibody, let's talk about what is meant by monoclonal antibody. Monoclonal antibody are identical immunoglobulins generated from single B clone. They recognize single epitope. By epitope we mean part of the antigen molecule to which the antibody will attach itself. Monoclonal antibodies are produced in vitro, that is outside the body, unlike polyclonal antibodies which are produced in the live animals. I am not going into the details about how a monoclonal antibody is produced. If you are interested to know in much more detail, please let me know in the comment. I will make a separate video on monoclonal and polyclonal antibody. So now we know what is antibody and what is meant by monoclonal antibody. Let's now understand the basic idea behind this research paper. The scientists found that one antibody 47D11 showed neutralizing activity for both SARS-CoV that caused SARS in 2003 and the ongoing SARS-CoV-2 that is causing Covid-19 pandemic. This coronavirus neutralizing antibody targets the spike glycoprotein on the viral surface that mediates the entry into the host cell. The S protein has 2 functional parts, S1 and S2. The S1 subunit that mediates cell attachment has 4 core domains, S1A through S1D, and fusion of viral and cellular membrane is mediated by S2 subunit. The spike protein of both SARS-CoV and SARS-CoV-2 binds to the human SE protein as a host receptor through S1B domain. If you want to know a bit more details about how the SARS-CoV-2 binds to human SE receptor, please refer to my previous video. I will provide you the link in the description. So, the spike protein of both SARS-CoV and SARS-CoV-2 binds to the human ACE2 protein as a host receptor through S1B. This receptor interaction is known to trigger irreversible conformational change in the coronavirus spike protein, enabling membrane fusion. The scientists reformatted the chimeric 47D11H2L2 antibody to fully human immunoglobulin, and it was found that 47D11 targets a conserved epitope in SARS-CoV-2 S1B domain. Thus, according to them, this antibody will be useful for development of antigen detection test and serological assay targeting SARS-CoV-2. Neutralizing antibodies can alter the course of infection in infected host, supporting virus clearance, or protect an uninfected host that is exposed to the virus. Hence, this antibody, either alone or in combination, offers the potential to prevent or treat Covid-19. Meanwhile, according to the Israel Defense Minister Naftali Bennett, their monoclonal antibody can neutralize the virus within the bodies of those ill. According to them, the key parameters are: the antibody is monoclonal, new and refined, and contains an exceptionally low proportion of harmful proteins. The antibody is able to neutralize coronavirus. The antibody was specifically tested on aggressive coronavirus. Thus, they are very confident that their monoclonal antibody can be used for passive immunization of the ill patients. They are now looking for applying patent and then go for mass production. However, this can only be done after obtaining requisite approvals from regulatory agencies for therapeutic use in patient after sharing results of human clinical trial. Thus, it seems like a natural extension of convalescent plasma therapy, and it has shown promising results so far. So that's it for today, guys. Hope you have found this information useful. Please hit the like button if you like the video and subscribe to my channel for more such videos. Thank you.